Direct DNA transfer to plants. And topic is protoplast transformation. Initially, we focused at how some biological systems that are based on agrobacterium tumefacients, how they can be used for plant transformation. And here we are going to focus that how plant cells can be transformed by using some direct methods so that plant cells can directly uptake the DNA from its surrounding environment. We have the idea that if the plant cells they are lacking cell wall, then the remaining structure that is left behind, it is called as protoplast. And protoplast can be transformed directly by using different techniques. प्रोटोप्लास्ट ट्रांसफॉर्मेशन जो है ये मोर और लेस हम कह सकते हैं कि जो एनिमल सेल्स की ट्रांसफॉर्मेशन है जिसको हम ट्रांसफेक्शन का नाम देते हैं उससे ये ज़्यादा रिजेंबल करती है. इन केस ऑफ प्रोटोप्लास्ट ट्रांसफॉर्मेशन exogenous DNA it can be incubated with protoplast and then it can result in the uptake into the nuclear DNA where it can be integrated. इस किस्म की जो ट्रांसफॉर्मेशन है इसमें हम कुछ केमिकल्स की भी हेल्प ले सकते हैं जो कि मीडिएट करते हैं ट्रांसफॉर्मेशन के प्रोसेस को. मिसाल के तौर पर जो पॉली इथाइलिन ग्लाइकोल है जिसको हम PEG का नाम देते हैं इसको यूज़ किया जा सकता है जो प्रोटोप्लास्ट की ट्रांसफॉर्मेशन है उसको इन्हेंस करने के लिए या मीडिएट करने के लिए. इसके अलावा जो एक और टेक्निक है जो कि बड़ी कॉमनली यूज़ की जाती है उसको इलेक्ट्रोपोरेशन कहते हैं कि अंडर इलेक्ट्रिक करंट्स जो प्रोटोप्लास्ट में जो टेम्परेरी क्रैक प्रोड्यूस होते हैं उससे DNA जो है वो न्यूक्लियर जीनोम के साथ जाके इंटीग्रेट कर जाता है. तो जैसा कि बैक्टीरियल सेल्स में है या एनिमल सेल्स के लिए हमें कुछ सिलेक्टेबल मार्कर चाहिए होते हैं तो इस तरह से जो डायरेक्ट DNA ट्रांसफ़र है here we also need some sort of selectable marker like antibiotic resistance. In this case, the selectable marker gene or the gene of choice or exogenous DNA, it can be ligated on the same vector or it can be co-segregated or co-transformed with the help of two separate plasmids. No transformation is there, and to confirm it, here we are going to use the selectable marker, that is some sort of antibiotic resistance traits can be used here. For this purpose, transformed protoplasts they can be grown on a suitable selectable medium where only those protoplasts will grow that contain the foreign exogenous gene along with some selectable marker. On an artificial media, protoplast, they will synthesize new cell wall and then later on the cell division will start that result in the production of a mass of undifferentiated or disorganized cells that we call it as a callus. And then this callus tissue it can be used to generate root or shoots by using another culture medium that contain an appropriate balance of different type of phytohormones. Now dicots, protoplasts, they have been frequently used for transformation and stable transformation has been carried out and different examples of dicots are there. But for monocots, there is a problem that some direct transformation methods like that may be based on agrobacterium tumefacients, they are not so effective or the transformation is difficult. And here the monocots, protoplast can be directly transformed by using their protoplast. So although some other limitations are there because monocot cotyledonous plants they are resistant to certain antibiotics like the kanamycin that are frequently used for dicots. So for this purpose, some other antibiotics were selected for the regeneration of transgenic lines from protoplast cells. Overall, we can say that protoplast cultures, they can be used for the regeneration of the transgenic plants.